Take a moment and think about what it felt like the first time you fell in love. The butterflies, the intense attraction, the warm, fuzzy feelings. Now imagine the crushing grief, the stress, and the anxiety of losing the person you love the most. These complex emotions are a quintessentially human experience, right? Well, actually, no. Scientists have discovered that the same chemicals and neural circuits that make us feel hopelessly in love or stricken with grief also operate in extremely similar ways in the brains of animals who form lifelong pair bonds. I'm going to start with a real-life story of two Canada geese that illustrates just how strong the bond between non-human partners can be. But this video isn't about cute animal stories. It's about the unsettling scientific evidence that many non-human animals love in much the same way that we do, and what this means for how we treat them. A few years ago, something happened at a wildlife veterinary hospital in Massachusetts that challenged a lot of people's preconceived notions about the emotional capacities of non-human animals. The New England Wildlife Center in Barnstable, Cape Cod, overlooks a pond that's home to some Canada geese. And in 2021, staff at the center noticed that a male goose, who they called Arnold, was limping severely and struggling to walk. They captured him to see if they could treat his injury and discovered that he had 2 open fractures on his foot, so they brought him into the clinic for surgery. And as they were prepping him for surgery, they heard a tapping on the glass door. It turned out that Arnold's mate, who they called Amelia, had discovered where they had taken Arnold and was trying to get inside.
Don't worry, he's in good hands. Ma'am, don't worry, we're going to do everything we can.
This is our first time we've ever had a patient try to visit while the other patient is in care.
Right now he's in surgery, and we'll let you know how it goes.
So we did the whole surgery while she was watching the whole thing. She stood literally without moving. I can only imagine what she thought was going on because he's asleep on the table at this point. When he was waking up from anesthesia, these are wild animals, it can be a stressful moment. So we decided that we would give Arnold his oxygen right there on the floor next to the door. And so we opened the door and him and his mate were there. And you could tell that she calmed down right away and he was also pretty calm. She actually stuck her neck in and started to groom him while he was waking up. It was very nice.
While Arnold was recovering at the center, the staff would put him in an outdoor pen for an hour each day. And every time they did, without fail, Amelia would come running and stay with him in the pen. This went on for a month until Arnold eventually made a full recovery. And when he was released into the wild, Amelia immediately joined him and they returned to the pond together. Now, if that's not a heartwarming story, I don't know what is. But scientifically speaking, can we actually say that Arnold and Amelia are in love? Or is that just describing human emotions to them? Is there any way that we can know, or at least make an educated guess about what animals like Arnold and Amelia might feel for each other? That's what we're going to talk about in this video. If you're new to this channel, my name is Dr. Mickey Pardo. I'm a biologist with a PhD in animal behavior from Cornell University, and I've been studying animal behavior for over 16 years. On this channel, I talk about the science of animal cognition and its implications for how we relate to other animals. And in this video, we're going to be tackling the question of whether any non-human animals are capable of feeling romantic love, or if this is an experience that's unique to humans. We're going to get into some hardcore neuroscience in this video, but before we do, a bit of background. About 90% of all bird species, a minority of mammal and fish species, and even a few reptiles, amphibians, and invertebrates form monogamous pair bonds. In some species, these bonds only last one breeding season, while in others, like Canada geese, the bonds often last a lifetime. Behaviorally, a pair bond can be defined as a relatively long-lasting sexual relationship between two individuals who show a preference for interacting with each other compared to other members of their species. In many species with pair bonds, both parents work together to raise their offspring, although this isn't universally the case. And while pair bonds are usually between a male and a female, this is also not always the case. Committed same-sex pair bonds are actually more common than you might think in nature, especially in birds. One of the most important defining features of a pair bond is the mutual preference component. In studies of pair bonding in non-human animals, one of the main ways that researchers determine the two individuals have formed pair bond is that they prefer to be with each other compared to a different member of the opposite sex. By far, the best studied species when it comes to the neuroscience of pair bonding is the prairie vole. Prairie voles are a species of rodent native to North America that form long-lasting, often lifelong pair bonds, and they form these pair bonds very quickly. In a lab setting, if you put an unpaired male and female prairie vole together, typically they'll have sex multiple times over the next several hours and start cuddling together. And after 24 hours, they'll show a clear preference for spending time with and cuddling with each other compared to other prairie voles, indicating that they've formed a pair bond. On a neurological level, the sex causes a part of the brain called the hypothalamus to release the hormone oxytocin, and in males, the hypothalamus also releases the hormone vasopressin. The oxytocin travels from the hypothalamus to two brain regions called the nucleus accumbens and the ventral tegmental area, and binds to receptors there. When oxytocin binds to receptors in the ventral tegmental area, this causes neurons that project from the ventral tegmental area to the nucleus accumbens to release the neurotransmitter dopamine into the nucleus accumbens. And then this dopamine interacts with oxytocin that went to the nucleus accumbens to create a rewarding feeling associated with the vole's partner, which helps cement the pair bond. There are actually two types of dopamine receptors in the nucleus accumbens: D1 receptors and D2 receptors. And it's specifically the D2 receptors that facilitate the formation of pair bonds in prairie voles, which is an important detail that will come up a few more times in this video. Neurons from the ventral tegmental area also send dopamine to another brain region called the medial prefrontal cortex. And the medial prefrontal cortex, in turn, regulates the nucleus accumbens and the ventral tegmental area, and is thought to be important for associating the rewarding feeling generated by oxytocin and dopamine signaling with sensory cues like the smell of the vole's partner. In male prairie voles, as I mentioned, the hypothalamus also releases the hormone vasopressin in addition to oxytocin. This vasopressin binds to a type of receptor called the V1A receptor in brain regions called the lateral septum and the ventral pallidum, and this is crucial for pair bonding in male prairie voles in addition to the oxytocin and dopamine signaling that I already discussed. Now, this is an oversimplification of how pair bonds form in prairie voles, but these are some of the major components of the process, which scientists have figured out through decades of highly invasive experimentation. For example, the way we know that oxytocin in the nucleus accumbens is important for the formation of pair bonds is through experiments where scientists surgically implanted a tube through a prairie vole's skull and into their nucleus accumbens. Then they used this tube as a guide to inject a drug that blocks oxytocin receptors into the nucleus accumbens, and saw that blocking these receptors prevented the prairie voles from forming pair bonds. Then finally, they killed the voles and dissected their brains to make sure that the tube actually went into the nucleus accumbens instead of a nearby area. And this is pretty typical of research in neuroscience. Most studies on any kind of fine-scale cellular or molecular process in the brain are at the very least going to require performing invasive brain surgery on your subjects and then killing them at the end of the experiment. Depending on the research question, experiments also frequently involve socially isolating animals, intentionally subjecting them to chronic stress, or destroying parts of their brain or sensory organs while they're still alive. And in cases where anesthesia drugs would interfere with the biochemical processes being studied, neuroscientists sometimes cut animals' heads off while they're still fully conscious. Since humans have legal rights, you can't do these kinds of experiments on them, so it's much harder to study the neurobiology of pair bonding in humans. But the evidence we do have suggests that the mechanisms are very similar to prairie voles. For example, in one study, men were given a dose of oxytocin, and then they were asked to look at photos of their own partner and another woman while researchers scanned their brains with an MRI machine. The men who were given oxytocin rated their partner's photo as more attractive compared to the men who were given a placebo. And when they were looking at their partner's photo, the MRI scan showed increased activity in the nucleus accumbens and the ventral tegmental area, the exact same brain regions where oxytocin binds to facilitate pair bonding in prairie voles. Another study found that the gene responsible for making the oxytocin receptor is turned on in the nucleus accumbens of humans, but not in rhesus monkeys who don't form pair bonds. In both humans and prairie voles, individual variation in the oxytocin receptor gene is associated with pair bonding behavior. For example, people who have certain variants of the oxytocin receptor gene are more likely to have marital problems. And the same is true for men with regard to vasopressin, which is the hormone that's critical for pair bonding in male prairie voles. Men with certain variants of the gene for the V1A vasopressin receptor are more likely to have marital problems and less likely to have high-quality romantic relationships. Finally, just like in prairie voles, dopamine seems to play an important role in human pair bonding. One study used PET scans to track dopamine in the brains of human volunteers who were in the early stages of a romantic relationship. When volunteers were looking at a picture of their romantic partner, the PET scan showed more dopamine binding in the medial prefrontal cortex compared to when they were looking at a photo of a platonic friend. And as I mentioned earlier, dopamine signaling in the medial prefrontal cortex is also important for pair bonding in prairie voles. Not only are the neurological mechanisms of pair bonding very similar across mammals like prairie voles and humans, but they're also strikingly similar in birds and fish. For example, when zebra finches form a pair bond, their hypothalamus releases the bird equivalent of oxytocin. And when scientists injected zebra finches with a drug that blocks oxytocin, this reduced their ability to form pair bonds. Dopamine is also important for forming pair bonds in zebra finches, and just like in mammals, experiments have shown that it is specifically the D2 dopamine receptors that are involved in pair bond formation in zebra finches, not the D1 receptors. Finally, the bird equivalents of vasopressin and its V1A receptor affect pair bonding behavior in male zebra finches, which parallels the way that vasopressin affects pair bonding in male prairie voles and human men, although the details of exactly how this hormone affects male pair bonding behavior differ somewhat between zebra finches and prairie voles. Similarly, in convict cichlids, a monogamous species of fish, unpaired males who were injected with a drug that blocks the fish equivalents of oxytocin and vasopressin showed reduced courtship behavior towards females. Another study in oval butterfly fish found that pair-bonded individuals had more dopamine receptors in the dorsal nucleus of the ventral telencephalon, which is the part of the fish brain that essentially corresponds to the nucleus accumbens in mammals. So that suggests that the same neurochemicals and brain regions are important for pair bonding across vertebrates, from prairie voles to humans to fish. Once a pair bond has formed, both partners work together to help maintain their bond. One way that many animals do this is through physical touch. So for example, titi monkey couples will sit next to each other and twine their tails together to reinforce their bond, which is similar to the way that human couples hold hands. Other animals maintain their bond through elaborate courtship displays or duets that require a high degree of coordination. Coordination between partners is very important for being effective parents, especially in species where the male and female take turns incubating eggs, feeding the young, or carrying them around. And there's evidence that animal couples who are more in sync with each other are more likely to stay together and more likely to have surviving offspring. In humans, obviously an important component of romantic relationships is that partners support each other emotionally, and the same is true in non-human animals. Studies on a number of species have found that just being with their bonded partner helps reduce animals' stress on a physiological level, and animals often actively engage in behaviors to console their partner when they're in distress. For example, when prairie voles are stressed, their partner will groom them to help them calm down, just like we saw Amelia the Canada goose doing with her partner Arnold when he was recovering from surgery. And this has a measurable effect on the stressed individual's anxiety levels. Not only that, but when prairie voles see their partner in distress, a part of their brain called the anterior cingulate cortex becomes more active. This part of the brain is known to be involved in processing physical pain, which suggests that prairie voles literally feel their partner's pain. It's also the same part of the brain that lights up when researchers put human volunteers in an MRI machine and ask them to imagine their loved ones in pain. Another important component of maintaining the pair bond is neurological mechanisms that motivate individuals to stay with their partner. In many species, including prairie voles, zebra finches, and humans, there's evidence that continued oxytocin signaling throughout the relationship helps maintain attraction to one's partner. And at least in prairie voles, Dopamine actually plays an important role in maintaining pair bonds by decreasing attraction to strangers. Remember how I said earlier that there are two different types of dopamine receptors in the nucleus accumbens, D1 receptors and D2 receptors? D2 receptors are really important for facilitating pair bond formation, but D1 receptors actually have the opposite effect. They prevent pair bonds from forming. Right after male prairie voles have formed a pair bond, the density of D1 dopamine receptors in their nucleus accumbens increases by 60%, and this helps prevent them from forming a new pair bond with someone else. In fact, while unpaired prairie voles are usually quite friendly and gregarious, pair-bonded prairie voles will aggressively chase away members of the opposite sex who are not their partner. With that said, just like humans, non-human animals who form pair bonds do sometimes cheat on their partners or leave them entirely. The level of sexual fidelity within a pair bond is highly dependent on the species. For example, genetic data on parentage has revealed that Florida scrub jays almost never cheat on their partners, whereas superb fairy wrens are so promiscuous that up to 76% of chicks are fathered by someone other than their mother's partner. And just like in humans, there's a lot of individual variation in personality among members of the same species that affects their propensity to cheat. Animals often engage in mate guarding behavior to prevent their partner from having sex with someone else. For example, being highly aggressive to potential rivals or trying to physically prevent their mate from straying too far from them. And an obvious question is, what is going on in the animals' minds during this mate guarding behavior? Do they feel jealous like humans do? In humans, MRI studies have shown that jealousy is associated with a number of brain regions involved in processing fear, pain, and aggression. One region that's activated when we feel jealous is the anterior cingulate cortex, which, as we've seen, is associated with both physical and emotional pain. And a study on coppery titi monkeys found very similar results. Researchers put male titi monkeys in a cage by themselves, where they could see another cage that held either their own mate together with a male stranger, or a female stranger together with a male stranger. Then the researchers imaged the male monkeys' brains with MRI and PET scans, and when the male monkeys saw their own mate in a cage with a male stranger, their anterior cingulate cortex, that part of the brain associated with emotional pain, lit up more than when they saw two strangers in the other cage. In both titi monkeys and prairie voles, experiments have shown that D1 dopamine receptors are involved in aggressive mate guarding, and there's indirect evidence to suggest that the same is true in humans. For example, drugs that mimic the effects of dopamine are sometimes used as a treatment for Parkinson's disease, and one side effect of these drugs is that they can cause patients to experience extreme delusional jealousy. When pair-bonded animals lose their partner, they often exhibit behavioral and physiological signs of grief, including symptoms of stress, depression, and anxiety. Konrad Lorenz, who won the Nobel Prize in 1973 for his work on the behavior of greylag geese, wrote that they exhibit, quote, virtually the same symptoms of grief as humans. He observed that greylag geese who lost their partners would hang their head, lose their appetite, and become indifferent to stimuli from the environment. They also became more prone to accidents and were more likely to be killed by predators. If they survived, they usually spent a while being single before finding a new partner, and sometimes they remain single for the rest of their lives. When prairie voles lose their partner, they start producing more dopamine receptors in the anterior cingulate cortex and the insula, both of which are associated with pain and negative emotions. And humans who have lost a loved one also experience increased activity in these same brain regions. Another thing that happens when prairie voles lose their partner is that their hypothalamus releases a hormone called corticotropin releasing factor, or CRF. And CRF does two main things. First, it causes stress hormones to be released into the bloodstream. These stress hormones remain elevated for about a month, and during that time, prairie voles usually won't form a pair bond with anyone else. Second, CRF prevents oxytocin from being released into the nucleus accumbens. All the evidence we have suggests that the biochemical basis of grief is very similar in humans. Just like prairie voles, humans who have lost a loved one have chronically elevated stress hormones, And oxytocin likely plays a role in human grief as well, because people who have a certain variant of the oxytocin receptor gene tend to experience more severe grief following a loss. We saw earlier that the presence of oxytocin in the nucleus accumbens helps create a rewarding feeling that's important for the formation and maintenance of pair bonds. So you might expect that the sudden drop in oxytocin levels following a loss would result in the opposite of a rewarding feeling, in other words, depression, and that's exactly what we see in prairie voles. Just like bereaved humans, prairie voles who have lost their partner exhibit behaviors that are thought to be indicative of depression and anxiety, and invasive experiments have shown that this is specifically caused by CRF reducing the amount of oxytocin in their nucleus accumbens. Two tests that are commonly used to assess quote-unquote depression-like symptoms in rodents are the forced swim test and the tail suspension test. In the forced swim test, scientists put the vole in a beaker of water that they can't escape from and measure how long it takes until they stop trying to swim and just float listlessly. In the tail suspension test, they hang the vole upside down by their tail and measure how long it takes until they stop trying to right themselves. The idea is that the more depressed an individual is, the faster they'll give up and stop trying to save themselves. To measure anxiety-like symptoms in rodents, scientists use a variety of tests designed around the fact that rodents are naturally nervous about being out in the open. So for example, in the elevated plus maze tests, scientists put a vole on an apparatus that has 2 covered arms and 2 exposed arms. And the idea is that the more anxious the vole is, the less time they'll spend in the exposed arms of the maze. Studies have repeatedly found that prairie voles who have been forcibly separated from their partner exhibit increased depression-like and anxiety-like symptoms in these tests. Many other animals who form long-term pair bonds have also been shown to exhibit increased stress and negative emotional states after losing their partner, including convict cichlid fish, coppery titi monkeys, and zebra finches, just to name a few. In humans, the thing that distinguishes grief from other negative emotions is the sense of yearning for the lost relationship. You don't just feel sad in general, you specifically miss the person who's gone. And one study on male prairie voles found evidence suggesting that they miss their partner too. After the voles were forcibly separated from their partner, they spent significantly more time sniffing bedding that smelled like their partner, but not bedding that smelled like a stranger or like food, which suggests that just like humans, they're specifically motivated to search for their missing partner. So to bring this all back to the question that I posed at the beginning of this video, can any non-human animals feel romantic love, or is this an experience that's unique to humans? A lot of scientists are very reluctant to use the L word for non-human animals and prefer to stick with the term pair bonding. For example, a 2018 review paper by Hassa Valum and Larry Young argued that pair bonding is an essential component of love but is not fully equivalent to it. They defined romantic love as the emergent property of integrating pair bonding mechanisms with, quote, the ability to translate multimodal sensory information from our partner into the mental language of romance, the ability to reflect on past interactions with our partner, and the ability to contemplate our future life journey together. They claimed that these abilities are unique to humans because of the complexity of our cortex, the part of the brain involved in complex thought. Personally, I find this argument unconvincing for 2 reasons. First of all, it may not even be true that these abilities are unique to humans. As far as I'm aware, no one has even tried to design experiments to test if non-human animals can reflect on their past and future with their partner. But there's growing evidence that many non-human animals can in fact think about the past and the future in at least some contexts. I actually made a YouTube short about how California scrub jays plan for future meals, which I'll try to link to up here. Up here? One of those. And if they can plan for the future in some contexts, it might not be too much of a stretch to think that they could imagine their future with their partner. Second of all, I'm not sure that the ability to reflect on past interactions with our partner and imagine our future together is really the defining characteristic of love as most people think of it. It seems to me that love is really defined by the feelings we have towards our partner in the present, and all the reflection about the past and the future is just icing on the cake, so to speak, that's not really fundamental to what makes love love. But whether we choose to call it love or not, I think the evidence is pretty overwhelming that many pair-bonded animals have intense, enduring feelings for their partner. As we've seen in this video, the same hormones, neurotransmitters, receptor molecules, and brain regions are involved in pair bonding, jealousy, and grief in humans and other animals. Just like humans, non-human animals provide emotional support to their partner and feel their pain when they're in distress. And when they're separated from their partner, just like humans, they exhibit behavioral and physiological symptoms of depression and anxiety, and a strong motivation to search for their missing partner. Personally, I think it would be more parsimonious to use the same term for this that we do for romantic love in humans, rather than trying to create a semantic distinction between humans and other animals that probably isn't biologically meaningful. In other words, while we can never be 100% certain about what another animal feels, I think it would be more reasonable to conclude that many non-human animals can feel romantic love than to conclude that they cannot. In my opinion, there may also be a darker reason why many of my fellow scientists are reluctant to say that non-human animals feel love. If we acknowledge that their feelings are similar enough to ours to qualify as love, what does that say about the morality of subjecting them to highly invasive surgeries, forcibly separating them from their partners, and killing them in order to study how these feelings work? There's this sort of mental tightrope that scientists in this field have to walk, where on the one hand, pair bonding in non-human animals has to be pretty similar to human love, or else there would be no point in studying animal models to learn about the neurobiology of romantic relationships. But on the other hand, researchers have to believe that their subjects aren't too similar to humans so that they can feel okay about doing experiments on them that they would never perform on a human. And one way to deal with that cognitive dissonance on a subconscious level is to use different terms for what is, in fact, extremely similar behavior in humans and non-human animals. But the terminology we use doesn't change the reality of what the animals experience, and I think we need to take a long, hard look at whether these kinds of experiments should still be legal, especially in light of what they have revealed about animals' emotional capacities. One final note. The ethical implications of the research on pair bonding go far beyond the scientists who do these experiments. For example, debates about the ethics of hunting usually center around the impact on the individuals who are hunted or the impact on wildlife populations. But one thing that rarely seems to factor into these debates is the impact on the individuals who are left behind. Many species that are legally hunted form long-term, often lifelong monogamous pair bonds, including most species of geese, swans, cranes, coots, doves, and pigeons, some species of ducks, quail, and partridges, most wild members of the dog family, including foxes, coyotes, and wolves, many small antelope, including dik-diks, duikers, and klipspringers, and some rodents like beavers and agoutis, just to give a few examples. It's extremely likely that these animals experience a great deal of psychological suffering when their partner is killed. So if you hunt any of these species, or others that form these kinds of bonds, even if you manage to kill the individual you're after quickly and painlessly, you're still subjecting their partner to long-lasting, painful feelings of grief. Hopefully this provides some food for thought. Thanks for watching, and see you next time.